Okay, so welcome to this fifth video on the phosphoinositide pathway. In this video, what I want to discuss is a little bit of the history of the phosphoinositide pathway. So basically, in 1953, two people by the names of Mabel and Lowell Hokin, sorry, that should be Mabel and Lowell Hokin, did experiments into the phosphoinositide pathway. And basically, what they were doing is they were stimulating muscarinic receptors with acetylcholine. So they were using acetylcholine to stimulate muscarinic acetylcholine receptors. And these basically were linked to G proteins, and therefore led to the activation of phospholipase C beta 2 in the way that we've discussed in the previous videos. Okay, phospholipase C beta was breaking down phosphoinositide phosphatidylinositol 4,5-bisphosphate (PIP2) into IP3 and diacylglyceride. And basically, IP3 was then leading to a calcium release from the endoplasmic reticulum by activating the IP3 receptor so that it can then be activated to open by calcium. Okay, so at this time this pathway was not understood, and what Mabel and Lowell Hokin did was they noted that if you put radio-labeled phosphate groups, so radio-labeled phosphate groups into your cell, which just means that you take the phosphate group, so this is the phosphate group here, and you put radio-labeled phosphate groups into your cell, and you then stimulate it with acetylcholine to stimulate the muscarinic acetylcholine receptors. What Mabel and Lowell Hokin saw is they saw these phosphate groups, well, they saw this radioactively labeled phosphate groups were going into the membrane. So when they stimulated this pathway with acetylcholine, what they saw is that the radio-labeled phosphates were going into the cell membrane afterwards. So why was that? Well, basically, when you stimulate this pathway, you stimulate the breakdown of PIP2. And now, PIP2 is too low in the cell membrane. So what has to happen? Well, you have to rebuild the PIP2 through the pathway that we have talked about at length in the previous video. I.e., you have to degrade IP3 gradually to inositol. You then combine inositol with phosphatidate to get phosphatidylinositol, and then what you have to do is you have to then gradually phosphorylate the 4th carbon hydroxyl group and then the 5th carbon hydroxyl group of inositol to return it back to PIP2. And these phosphorylation steps are going to add phosphate groups onto this molecule. And if you have put in radio-labeled phosphate into the cell, then these radio-labeled phosphate groups may well be added onto these phosphatidylinositol molecules to create the phosphatidylinositol 4-phosphate and then the phosphatidylinositol 4,5-bisphosphate, and therefore the radio-labeled phosphate would end up in the membrane. So that was the first sort of inkling that these pathways involved these phosphoinositides, basically. That there was something happening with this phosphate group, these radio-labeled phosphates, that was then leading them to go into the membrane. Okay, so that's what Mabel and Lowell Hokin noticed. Now what we're going to turn to is a little bit later, in 1983, Mike Berridge managed to get a grip on whether the phosphoinositide response, which this acetylcholine was stimulating through this muscarinic acetylcholine receptor, whether that preceded the calcium response, because the IP3 receptor, by the way, is going to lead to calcium going up in the cytoplasm, and basically it was controversial as to whether the phosphoinositide response here came before the calcium response or whether it came after the calcium response, whether it happened because of calcium going up. What was known is that when you stimulated with acetylcholine, calcium went up, and also there was this phosphoinositide response that was illustrated through these phosphate groups being being accumulated in the membrane. So the guy who really gave us information on whether which came first, basically, was a guy called Mike Berridge. And basically what Mike Berridge did is he used a fly. He used a fly known as the blowfly. So blowflies have a rather unusual portion of their physiology. They have a salivary gland which actually secretes IP3. And now this is very, very interesting for those people studying IP3, the phosphoinositide pathway, because let me explain to you how this salivary gland works in this blowfly. So basically, what happens is if this is a cell on the salivary gland of the cell of the blowfly, which is going to secrete saliva, basically, then it's stimulated to start secreting saliva by 5-hydroxytryptamine, or serotonin, basically. Tryptamine. So 5-HT, or 5-hydroxytryptamine, or serotonin, or whatever you want to call it, this molecule comes and binds to receptors on the basolateral surface of this cell. And basically, through those receptors, it's going to activate both cyclic AMP to go up through the GS coupled G protein coupled receptors, and it's also going to activate the phosphoinositide pathway, basically. So it's going to make IP3 go up, and then it's going to, IP3 going up is going to cause calcium to go up. And basically what happens is that cyclic AMP activates potassium channels on the apical surface of this cell, like so. So potassium is going to be secreted, and calcium activates chloride channels on the apical surface of this cell. So here's a chloride channel. So chloride anions are going to be secreted, like so. Okay, so these two responses are overall going to lead to potassium chloride being secreted, and that is then going to mean that we've secreted salt. So you're then going to get the movement of water following the salt. So water will move by osmosis, and you'll produce us a nice saliva out here. Now, what's unusual is that these blowflies actually also lose the IP3 in this saliva. So IP3 is also going out into the saliva. Now that's very, very useful, because that means that the IP3 cannot then be degraded and recycled. So we can't get this degradation to inositol and then the recycling into PIP2. So what's going to gradually happen to our blowfly salivary gland is that it's going to run out of PIP2. And then if you continue stimulating it with 5-hydroxytryptamine, you're going to get the calcium response going down, because you're not going to be able to create any more IP3. So eventually what you will see is that the calcium response no longer can happen. And the reason for that was because the IP3 had all gone and the phosphoinositide response was no longer happening. So through that, what Mike Berridge was able to show was that the phosphoinositide response preceded this calcium response, basically, and in fact triggered it and was essential for triggering it. So what he did indeed find was that if you continually stimulate the blowfly salivary gland with 5-hydroxytryptamine, eventually this calcium response stops, and it's because this phosphoinositide response runs out, basically. You run out of PIP2 in the membrane of these cells. And he was able to show that if you replenish the PIP2 in this cell, then you can restore the calcium response. And that really nailed it in people's minds, that the phosphoinositide response preceded the calcium response and was responsible for the release of internal calcium stores.